Today, I'm really honored to give this talk on the journey with the zebrafish to conquer blood disorders. Of course, when did my journey start and how I started this journey, I'm going to give you a glimpse of that and subsequently move on to the real science. When I was at the Indian Institute of Science, I thought of looking at the structure of an RNA called 5S ribosomal RNA. At that time, Carl Woese proposed this particular model of the so-called ACGU sequences, which is about 120 nucleotides. And I proposed this model after Carl Woese in 1975. This was done in 1977. Subsequently, I moved to Yale, where Professor Sherman Weissman was in fact known for the 5S RNA sequence from human cells. That is why I chose this laboratory. But at that time when I went there was a lot of students and postdocs who were sequencing these genes called beta globin genes and delta globin genes. But I did not want to sequence the genes, and I told him I would rather sequence in between genes called intergenic DNA, and in fact, it was also called junk DNA. So I started sequencing them, and lo and behold, I discovered repetitive DNA members called ALU families in human genome, which is about 1 million copies of 300 nucleotides and so on. Therefore, approximately quite a number of millions of base pairs would have been sequenced at that time, but that is not the case. Subsequently, I moved to University of Illinois College of Medicine, somewhere on the 11th floor. And I did not want to pursue what I learned at Yale. There's hemoglobin sequences and hemoglobin disorders. I chose a completely different path, and that is the blood coagulation pathways. These blood coagulation pathways are very unique, that there is a defense mechanism. When you start yourself injured or just cut yourself a little bit, the blood clots and prevents it from bleeding. In fact, it is like a dominoes. A series of factors activate one after another and the domino completes. But now it completes and plugs this wound. But if you have one cord, which is actually broken, this domino doesn't do that job, and essentially it will not seal the vessel, and thus the individual bleeds. When I started the program, there were no gene sequences available for clotting factors of human genes. Therefore, I decided to look at the genes like factor 9 gene and factor 10 gene. We isolated them. And in fact, the deficiency of factor 9 is called hemophilia. There are 3 types. One is hemophilia A, B, and C. A is called factor 8 deficiency, B is 9, and 10 deficiency, and so on. But then I was also interested in looking at the mutations in individuals suffering from hemophilia in Chicago area. And in fact, we isolated those mutants, but it was not satisfying in scientific endeavor. One of the mutations called factor 9 Leiden or hemophilia B Leiden, because the people who are suffering from hemophilia come from an area called Leiden in Netherlands. And these individuals have essentially 0% factor 9 levels when they are born. But as they age, after puberty, it starts more and more factor 9 levels. And then subsequently, the person gets cured of hemophilia A or of hemophilia B. So the question that I raised is, there is something, an engine is bad, but that engine is getting kick-started somewhere later on in life. And what is that factor that is actually activating the so-called gene which is dormant or inactive? So I decided to look at their mutations, and we found by the time I moved to University of Texas Health Science Center in San Antonio, and I looked at the mutations, and in fact, we found a mutation which is present in regulatory regions of the gene, not the coding sequences, but the regulatory area. So we took that regulatory area to solve the puzzle of which factor is binding to it and put a marker gene behind that regulatory area and put that whole DNA into a mouse, and that is called a transgenic mouse. This transgenic mouse expressed that marker gene very well as the mice aged. 8-month-old mice got almost like 60% activity and so on. But then I realized that I spent a lot of public money, NIH dollars, and I did not solve the puzzle at all. In fact, what I did was I recapitulated what was already known in humans. In humans, it said the fellow gets cured at the age of 40 or 50. The mouse got expression at the age of whatever, 8 months, comparable to what we have. I didn't solve that factor that is not there. So I thought it's going to be a very difficult task, because if I hypothesize there is X factor or Y or a Z or whatever the factor is, if I want to cripple that factor by knocking it out and see their effect, it is going to take me multiple of my lifetimes, which is impossible to do. Therefore, I was wondering what I should do. At that time, I visited France, Marseille, to an international transcribe workshop, where Sidney Brenner and his postdoc presented that he sequenced randomly 100 genes from the fugu fish, which is a puffer fish, and found that 80% of the genes, or 80 genes, are conserved or present in humans. Therefore, I thought, if I can take this fish and use them as a model like Drosophila, that mutate it and see how these genes are expressed in factor IX, etc., it would be a nice approach. But then quickly I figured out it is very difficult to culture this fish in the laboratory, and it is not a laboratory animal. Then I was looking for alternate model, and in fact, when I was searching for literature, I found this book, An Account of Fishes in the River Ganges and its Branches, published in 1822 by Francis Hamilton Buchanan. He is a medical doctor hired by Lord Wellesley, the Governor General of Calcutta, India, and he asked him to be his personal physician. But after 1 or 3 years or whatever it is, he fired him and made him Director of Forestry and Fisheries in India. Therefore, he goes and finds out 105 fishes in the river Ganges, indigenous ones, and one of them turns out to be a fish called Brachydanio rario. And this is now future zebrafish. And in fact, when it arrived to United States of America at the University of Oregon, as usual, the name has been shortened from Brachydanio rario to Danio rario, which is very usual in America. So I discovered this fish in 1995, and it was already popularized by George Streisinger in 1980s, where he in fact published a science paper. The front page says the cloning. The cloning was done later in the sheep dolly, but it was the first cloning which was done early on. What he did was he takes the male fish, mutagenizes them by a nasty chemical, crosses them with a female, and gets progeny which you can screen for the phenotype. What he was screening was a morphological phenotype. Like, for example, this embryo is splitting and giving rise to a blastula stage, etc., and gastrulation, subsequently involution, as you can see here, that this embryo goes through involution process and subsequently generates what is it called an eye, brain here, and a tail, and segments, as you can see. This only takes about 19.5 hours. I'm sure that women would love that if they can go through the pregnancy for 19.5 hours. So that was a very marvelous developmental program, but he was looking at only developmental biology. The reason is, as you can see, in this parking lot, if I ask you a question, how many red cars are there, you can figure it out. There is about 7 or 8 or 9, whatever that is. However, if I ask you the question, which car has the best engine or poor engine, then you have to open up the core, check those things, etc. That is a physiological, there is a functional one. And for example, our blood clotting, unfortunately, is a functional assay. That means it's a physiological one, which has been very difficult, unlike what George Streisinger was doing. So how do I use this model to solve the puzzle that I started for identifying this factor? Unfortunately, there was another person called Russell Doolittle at University of California, San Diego. He had a thorough thesis published in 1961 suggesting that the fish coagulation pathways are very primitive. He only said there are two factors, and therefore I said it is going to be difficult to really oppose him because it has been already a paradigm, and therefore I was just trying to begin to wonder whether I should believe Sidney Brenner because he said 80% of the genes are conserved. Therefore, I had an issue to do it at that time because he said only two proteins are there. I looked for other genes and cloned the genes for the human, corresponding to the human genes like zebrafish genes. There are 3 factor 7 genes, 2 factor 10 genes, and 2 factor 9 genes. Such a duplication was there in the fish. For example, humans have 25,000 genes, fish have about 30,000 or 35,000 genes. So then I cloned all the genes in that pathway and showed, in fact, the entire clotting cascade except for 2 factors which are missing here in the fish. Also, another challenge began. The challenge was that these fish cells on which these factors play, essentially dance on the surface of the cells, are thrombocytes, which are nucleated, whereas the factors which dance on cells in humans are called platelets, and those platelets are not nucleated. And there was another opposition telling that, after all, you are studying wrong cells. You are not studying the real cells of the fish. So therefore, we had again shown that all the receptors on the surface of these cells are very, very similar. And subsequently, we were also facing another challenge, that the assays, that the tests for blood clotting are missing in this fish. And even for that matter, even the large fish blood clotting assays were poorly developed. The challenge was that for each test of blood clotting, it requires about 200 microliters, almost like 4 or 5 drops of blood, whereas the fish entire body volume is only 50 microliters or a drop of blood. So we developed microassays utilizing only 2 microliters of blood that we could collect. And now we have approximately 30 different assays or tests to study this blood clotting. Interestingly, I could study the clotting. Suppose if I want to test blood clotting, I could use one of these assays and look for the mutations which will affect this particular assay. But that is called clotting assays. But unfortunately, if the clotting happens within inside the body, you call it thrombosis. If it happens outside, it protects you, whereas if it happens inside, it kills you. And therefore, we don't have an assay for thrombosis. We got a lot of assays for clotting, but we don't have an assay for thrombosis. Then I was thinking, I got this idea. I lived in Madras. It's now called Chennai. In the streets of Madras, the clotting occurs. I just want to show you this slide. For example, that is one of the vessels. This is another vessel. There is some kind of an insult or injury happens right here, and all these clotting occurs right in this area. You can see, watch that. There are small neutrophils, platelets, everything else is moving along. But after some time, what happens is that this clot also dissolves. That is called fibrinolysis. That the fibrin that is forming a clot, it lysed completely, and as you can see, it reestablishes the blood flow. So I thought I should somehow mimic this program, and I set up an assay called a laser-induced thrombosis assay, wherein a laser box delivers this laser through this microscope, and a small microscopic slide. You put these larvae, which are about 3-day-old or 4-day-old larvae, and then subsequently injure their vessel by the laser beam. So as you can see, it essentially cell by cell by cell aggregates. These are thrombocytes, and blocks this flow of the blood, and that is called essentially similar to what you call it a heart attack. So if this kind of a clotting happens in a coronary vessel or supplying to the heart, you call it a very fancy term called myocardial infarction. If it happens in the lung, you call it pulmonary thromboembolism. If it happens in the feet, you call it deep vein thrombosis, and so on. So that is the arterial thrombosis. This is venous thrombosis. As you can see, the cells are clustering here, which is quite different from this particular thrombosis. And subsequently, after some time, it is also going to clot and stop the flow. So what we have is a time to occlusion. That is, in other words, starting from the injury of the site, laser beam injury, you can measure actual time taken for the clot or occlusion to occur. That is called time to occlusion, or TTO. If it's about 21 seconds for the venous, 75 seconds for the arterial. But now if there is a mutation in any of the genes or players in this game, whether it is the cells which are lining the blood vessel, or the fluid that is flowing called plasma, or cells, any kind of a gene that is affecting these pathways, it would result in either prolongation of this clot time. If it goes to 120 seconds, you call it, what is it called, a bleeding. If it goes shorter than that, you call it thrombosis. You could score like that. So we did that. By the time I came to University of North Texas, and we conducted that mutagenesis I told you, and we screened for this phenotype of either prolongation of the time to occlusion or shortening of the time to occlusion. We characterized several mutations, and these obviously needed lots of tanks. And this portion is actually brought from San Antonio. This is all built here, and now we have a fancy facility in the department. So we used what is it called a linkage analysis for mapping these mutants. What it is very simply, you've got several markers, 200 markers randomly located, and if the one marker is very closely associated with your mutation, that marker and the mutation travel together. That's the principle of the linkage. It is like shuffling the cards. When you take two cards and put glue between the two cards and shuffle them, these two cards will always travel together. That's the principle of the linkage. So you can find one marker which is close to that mutation, and then go ahead and walk through that particular area and isolate the gene. We did that, and we found one mutation in one of the proteins called G protein coupled receptors, and that is called GPR34L. A simple amino acid change has given rise to a bleeding phenotype. But then I realized I almost spent about 2 decades to characterize these pathways, to establish the methodologies to really screen for these bleeding phenotypes, and I could get only one factor isolated. At that rate, I think I would need again multiple lifetimes to solve this puzzle, because there was another couple of groups have published that 10% of the genes may play a role in a major pathway. For example, thrombosis is a major pathway. It contains cells, plasma, endothelium, everything else. And 10% of the genes means we have got 25,000 genes in humans, approximately 30 from the fish. And if you compute that, you're going to get about 2,500 genes that may play a role in thrombosis. How do I get this 2,500? For the past 5 decades, 300 laboratories working across the world have solved about 300 factors so far. How am I going to discover these 2,200 factors, among which I should get that factor 9 controlling gene? That is the goal. So the bottom line is that previous methodologies are very inefficient, and therefore I came up what is it called a targeted knockdown by antisense morpholinos. These are modified nucleotides which will block the translation of the messenger RNA. So what we do is we inject this kind of a morpholino right into these embryos, and then look for its bleeding phenotype, either by time to occlusion or by looking morphologically whether it is bleeding. For example, here is an intracranial bleeding. There are a lot of other bleeding with the 2 known factors. But at this rate also, it is difficult because each person has to take injection needles, inject these reagents right into this area, it is going to be very, very difficult to do the 25,000 genes knockdowns one at a time. Therefore, we said, let us just modify this, that which can suit to the adult fish. Then we found another reagent where exactly the reagent is the same, except there is a bulky group which is attached to this morpholino group, and that one will make drill in the cells, go through the cells and penetrate and deliver this one, and subsequently block the protein synthesis. When we did that with a known gene, we got bleeding fish blood very well. But now the problem is, I just will show you how we inject this particular fish. For example, we are going to inject about 5 microliters of the reagent. The reagent is taken, put on this parafilm, just to make sure that we can suck this in the needle. I mean, for example, you can only suck only in that part of the needle, but not overall. So you got to go back and forth to make sure that it is really possible to deliver it. Then catch hold of the fish, put that fish in such a fashion that you put it under a paper towel or a tissue paper so that it doesn't, it doesn't see you, otherwise it will jump. So you cover the eye, rub it up, and of course you just use the third stripe from the top, that is the central blue stripe, right there you inject. If the injection worked very well, then when you take the needle back, you're going to have, what is it called, a blood coming out of that. So the injection is successful. Now you return this fish back into the water. The fish is happy, although it may not be happy, but so you get the fish injected. Like that, we could inject many of them, but unfortunately, the reagent costs are enormous. For one gene, it will cost you approximately $1,000. That is because they're sending you so much reagent, out of which you only use 1 or 2%. Rest of it is a waste. They earn the money, we lose the money. So the bottom line is, how do we definitely reduce the cost for this methodology? Therefore, we thought of an idea that if I take 1% of the VMO, or small amounts of that, and synthesize another reagent called antisense oligonucleotide, you can hybridize them, or you can just make a complex between the two. And this particular thing is the so-called viomorpholino, which is that bulky group I showed you, is going to piggyback this oligonucleotide into the cell, and then this portion of that red bar is going to complex with the messenger RNA, and that messenger RNA can be degraded by what is it called, another enzyme called RNase H. So this worked like a charm, like in some fishes, essentially 100% knockdown of the expression, and therefore we were happy that this is going to work very well. We wanted to test this. We chose a group of genes called serine protease genes, about 179 of them, knocked each one of them down, and found 2 important factors which were never being thought of as a part of the blood coagulation cascade, and that is called prostasin and hepatocyte growth factor. These turned out to be regulators for factor 7A production. Subsequently, now that we know that we can do many of them, we wanted to take another group of genes called homeobox genes, or called Hox genes, which usually are very important in segmentation as well as the pattern of the body. And when we discovered at least 4 of them reduced the number of these thrombocytes or the platelets, and 1 of them increased that particular number. So now that we can do this kind of analysis, we were a little bit greedy about it and said, can we piggyback 3 independent oligonucleotides? Therefore, what we did was we complexed them like this. And of course, lo and behold, it can go and bind to 3 different messenger RNAs and knock them down. So now this became a trivial undertaking. Therefore, we thought we could do glycosyl transferases, approximately 234 of them. These enzymes will essentially decorate or sugar coat the so-called factor, which is a pretty large factor, which is synthesized by a complex series of steps. It's a sugar-coated one. And when we knock these down, we could measure different levels of von Willebrand factor. Some of them reduced the levels, some of them enhanced the levels, and thus we found about 16 genes which are actually controlling the levels of the von Willebrand factor. When we injected, just combined 4 or 5 of them here, You see, you can see the bleeding spots right away, spontaneous bleeding in all the 4 fishes, exactly in more or less in the same location. So the technique is working very well. Then we said, let us ask another question of chromatin modifying proteins. For example, in our cells, the DNA is not naked. It is decorated by lots of proteins called histones and other molecules. And some of the proteins are modified by some other enzymes called either acetylases or methylases, etc. Those things also gave us interesting result that the chromatin modifying proteins, one of them called SUSY12B, when it is knocked down, one of the factors increased in its amount. This factor turns out to be an anticoagulant factor. Therefore, if you increase that, you're going to bleed. And that is exactly what we observed. Indeed, this particular protein called SUSY12 is actually is a big complex, a part of the big complex, which actually modifies these chromatin. So now there is a chemical which we identified that it is a FDA approved cancer drug. When you use this drug, you are also going to chop this complex, and therefore you expect it to have bleed more. That means the time to occlusion is much more than what we expect. So in other words, for 22 years or so, we've only identified one factor by the old-fashioned techniques. By modifying these technologies and inventing new methodologies, we were able to identify at least about 35 novel genes in this pathway, which was remarkably faster than what we anticipated. Now, coming back to these factors, when we discover them, we just want to have a fish not just purely knocked down, but we want to have a fish alive, which we can analyze for a longer period of time. Therefore, we wanted to see the CRISPR-Cas9 knockouts of all these factors. But before doing that, we just chose some known ones like von Willebrand factor. We knocked it out by the CRISPR-Cas9. I'm sure you know well CRISPR-Cas9 methodologies because it's a very popular one nowadays. And another gene we knocked out. And now we have about 7 disorders: von Willebrand disease, hemophilia A, hemophilia B, Bernard-Silver syndrome, factor V deficiency, prothrombin deficiency, and TFPA deficiency. All of these deficiencies are present in our lab. And so these fish could also be used in the future for gene therapy purposes, as well as therapeutic protocols can be developed using these fish. So now coming back to factor IX, now it becomes a trivial undertaking because, as I told you, these are all transcription factors which are controlling during aging, and there can be in the genome, there are about 1,000 to 1,500 transcription factors. Now it's not a big deal because 1,500 factors knocking down is very trivial because you are going to do in the groups of 3 or groups of 6. So you're only talking about 500 factors, 500 knockdowns, which is very easily can be done by a graduate student. But before that, we wanted to show that, in fact, there is an age-dependent expression of the factor IX gene, and in fact, it is there. Therefore, we'll be able to finish this program sooner or later. So in summary, we introduced zebrafish to study the genetics of hemostasis and thrombosis. We identified several novel genes which play a role in hemostasis. More comprehensive screening, as just I mentioned, to identify the genes involved in regulation of clotting factors should shed light on thrombosis. In fact, each novel gene identified will lead to an independent field by itself. There is now a hope that we will resolve the mechanism of age-dependent regulation, which we started 40 years ago, of the factor IX gene in future. Once it is done, I think it is time to go in any direction we want. Opportunities are knocking at the door. At that time, I think I will just relax in future. And these are all the graduate students who are involved in the zebrafish work. These are the postdoctoral fellows. And these are the current graduate students. These are investigators from whom I received some help. These are investigators who received help from my laboratory. And these are our local collaborators, particularly Arthur Govan, hired me here to come to this campus. Thank you. 25 minutes? Yeah. Thanks.
All right, this is, thank you very much for an excellent talk. We have a couple of microphones here in the audience for our question and answer period. So if you have any questions that you'd like to ask Jag about his excellent talk, please feel free to do so.
Yes,
A non-scientific question.
it's all right.
Eureka moment 25 years ago. Do you still remember it? When you clone all those genes?
Yes.
And what was it like? Can you describe it for us?
Oh my God. The factor 9 was a really a miracle. I used to synthesize oligonucleotides. Nowadays, you can order them. The 19 nucleotides, we used to make synthesis. In those days, it was supposed to be finger tapped overnight. That means one student has to take shifts, and somehow we made this synthetic oligonucleotide, but keep on finger tapping. And somehow we got that and used that as a probe to screen a CD in the library, which was obtained from Savio Wu from Baylor College of Medicine in 1982 or 83. So when we saw this spot, there was only one clone out of almost like 100,000 bacterial colonies. One clone showed us a beautiful hybridization, and that really started the program. It's really a great moment because on that day we opened champagne bottle, whether it was good or bad, and that's exactly what happened. That is really a great moment in my life that kickstarted the whole program. Any other questions? Yes.
By this morpholinos last actually after injection?
The morpholinos usually stay about until 8 days. And our assays are done in 4th or 5th day. So 8 days they lost. Vivo morpholinos also do the same kind of a job. But whereas if you inject simple oligonucleotide, the nucleases will chew it up. Fortunately, because of the so-called steric hindrance by the bulky group, that is an octaguidin dendrimer, and therefore that will protect it from cleavage by the random nucleases. But once it complexes with RNA, as you know, RNA DNA hybrids are more stable. And therefore that stays there. But unfortunately, RNA's H will come, or fortunately, it will come and cleave that off, and therefore a messenger RNA is degraded.
Would it have mattered if you had chosen for your model venous clotting or arterial clotting or air?
In fact, it's a very good question. You cannot just choose one particular clotting because the venous thrombosis involves initiation by red cells, whereas platelets or the thrombocytes come a little bit later. In arterial thrombosis, the first cell that attaches to the subendothelial matrix is a platelet or a thrombocyte. So that is purely platelet rich. So if you're interested in thrombocyte pathways, I think arterial is better. But venous will also pick up some of the arterial sections because the platelets are involved but come a little bit later. So I would prefer using venous, and also it is easy to do, and it is 21 seconds is time rather than 75 seconds. So those are the advantages.
So I guess normally you would expect air, but since the fish is in the water, it's going to be a little bit different there.
And this question has been puzzling me for a long time. And that is why I think in the last seminar, there's a great man called Aristotle who wrote a book on history of animal history there. So in that, he started talking about comparative biochemistry of blood coagulation of different animals. For example, he said turtles' blood clots the fastest. Fish blood clots about 6 times faster. And I don't know why he said that, but the roe and the deer blood clots very slowly. That's what he said, but that may be some other reason why he made a mistake. So because of that, the fish blood clots so rapidly, it clogs the wound in the water also very quickly, rather than in the air. That is my explanation. Yep. Why fish blood clots faster than humans is the question that I raised some time ago. The reason is that the so-called antithrombotic factor, the anticoagulant pathway is weaker in fish compared to anticoagulant pathway in humans. That is why it clots faster.
Does it give any advantage?
The selective advantage for the fish. I mean, obviously it has to clot fast, otherwise it'll keep on oozing out during swimming. So it has to clot faster. It will ooze, it will
hold
ooze.
your finger underwater.
You are
Also
right,
takes
you are right. In
longer.
fact, with that knowledge only, we also did, what is it called, collection of a blood. We cut the tail and then dip it in the buffer so that it drips into the buffer, so we can centrifuge the cells and find out the bleeding time. And after we have done this work at the University of Pennsylvania, 10 years later, or a few years later, they cut the mouse tail and dip it in the buffer so that it oozes much more, and they studied the bleeding time that way. So it is actually advantageous in one way to develop an assay, but it is advantageous to the fish. Yes.
Yeah, thank you so much, Professor Zhang. It's very fascinating talk.
Thank
And I do not have a background in the blood coagulation mechanism. Just I'm wondering, because there are many genes involved in that, like clotting and thrombosis. And in terms of future, like developing medicine or the drugs, how those challenges could be addressed?
For example, even in just one particular disease called von Willebrand disease, there are enormous number of variations in the patients, classified as a young, all sorts of different classifications are there. They're all because of the different levels of von Willebrand factor synthesized. Okay, nobody knows the mechanisms of those things. So if this kind of an approach, let us say with comprehensive screening identifies all the group of genes and puts in a bank 2,500 genes, then you can take all these patients. You don't have to analyze all the genes. You only have to focus on this 2,500 to identify mutations in the patients so that you can solve those puzzles. And that is one approach. And the second one would be to use these things for a therapeutic purposes. Once you identify the factor or protein, then you can molecular model some of the drugs that you are planning to do and see whether they interact with them. There is a lot more future for that. Any other questions? Thank you.
Thank you again, Jack, for an excellent talk. Thank you for coming to the Fall 2022 Smart Talk. I'd like to invite everyone to our reception, which will be upstairs in the President's Suite in room 406. We will have refreshments and an open bar for faculty, staff, and guests. Again, thank you very much for coming, and hope we'll see you in room 406.
Was it okay? Absolutely. Okay, thank you.
Lots of questions.
Yeah.
That doesn't always happen. That does not always happen. So
Thank you, thank you. So I need to, yeah.